Hello and welcome to another edition of this week's Labrats. So over the course of the last several weeks we've been learning about plants and the reason that we started with plants is because plants are a little bit easier to look at under a microscope than animal cells. Plant cells tend to be much larger. They also have a very obvious cell wall, and animal cells just don't have those things. They're a little bit harder to see under a microscope. So I wanted you guys to practice using a microscope and making slides before we started jumping into looking at animal cells under a microscope. So that's what we're going to do this week. We're actually going to look at our own cells under a microscope because I'm leading into teaching you about genetics, so about our own DNA and about extracting our own DNA. You probably are like, what's extracting DNA? Well, we're going to get to that and you're going to learn about it in a couple weeks. So this week you're going to need your microscope. So you're going to need some of your clean microscope slides, and you're going to need a cover slip this time. So these cover slips are just these plastic slips that you can barely, I'm sure you can't even see it in the video, but they're these plastic slips. So what you're going to want to do is you're going to want to take, I gave you plastic spoons. Ideally, you actually want a flat toothpick. So I didn't have flat toothpicks and I couldn't find them in town. So instead what I did for myself is I whittled down one of these skewers. So I just took, it and basically whittled it down. And what you're going to want to do is very gently, you're going to want to scrape the inside of your cheeks. So this experiment is best done before you brush your teeth, or if maybe at the end of the day, if you haven't brushed your teeth all day, because there's going to be more cells in your mouth that way. So the reason why we use the inside of the mouth is because your mouth actually sheds cells very quickly. So the reason it sheds them is because it actually grows them. So the cells in your mouth divide and grow faster than anywhere else in your body, and that's because there's obviously a lot of things happening in your mouth. That's where you eat, you have a lot of bacteria in your mouth, so your cells basically want to replicate and grow as fast as they can to keep your mouth healthy. So because of that, we get to fish some cells out of our mouth because there's lots of shells that are basically being shed off the inside. So we're just going to take this this little modified skewer here, and we're going to we're going to scrape the inside of our mouth. Try not to hurt yourself. Don't like put the stick through your cheek or something. Okay, and that might seem like you don't really have too much, but you'll have a bunch of cells. So what you're going to do is you're going to smear all that, and you'll actually kind of see it. When you smear it, it'll kind of look like there's almost this like kind of white film in it. Those are actually cells, so don't be entirely grossed out by that. That's floating around in your mouth regardless of whether or not you're noticing it or smearing it on a slide. The next thing you want to do is you're going to want to put a couple drops of food coloring. I gave you food coloring, but it's probably better to just run out and buy one, like one like this, so that you can actually kind of control how much you have. You're going to want to put a couple drops in a small dish, and you're going to want to have a dish of water. So what you're going to do is you're going to just take your pipette and you're going to draw up some of that food coloring, just a little bit like that. You don't need a lot, and you're going to put a drop of food coloring just like that on your slide. Okay, so then you're going to clean off your pipette, and you're going to take your cover slip for now. This is all we're going to do. We're going to take the cover slip, and we are going to just, at a 45-degree angle, I'll show you how to do this again. At a 45-degree angle, you just take it and slide it over until that cover slip is touching the liquid, and then you drop it. And that's the reason we do that, is because we don't want any air bubbles under our cover slip, right? If there's air bubbles, we can't see anything. It's the best way to keep air bubbles from getting under your slide. So we're just going to leave that. We're going to leave that for a couple minutes, and you can look at it under a microscope. If it's too dark, like if there's too much food coloring, what you can do, I'm going to show you a technique. You can just bring up a little bit of water here, and you can just drop it along the side of your slide like this. So just like this, and it's going to basically create a whole bunch of water underneath the slide. You can then take a piece of paper towel. Clearly I've already done this, and you can just dab it, and that paper towel is going to suck out some of that dye, and you're going to be left with a slide that has less dye in it. Okay, so you can see it a little bit better. This is a technique that we actually use in university to do this. So what you can do is you can just take that slide and put it aside for a couple minutes, because that food coloring needs to dye those cells, right? That's why we were using food coloring. It's not just to make it blue. It's not just to make it pretty. It's because that food coloring needs to, through osmosis, which we learned about, move into the cell to dye it a color, right? So we're trying to dye the cells so that we can see them better, because they're basically just invisible and transparent. They're not invisible, but they're transparent. So I've already done this. I've already made a slide. So what we're going to do is we're going to turn our light on in our microscope. We're going to put the slide under, and we're going to turn it to the lowest power, right? We're on the lowest power, and we're going to look through. And what you're going to see is a whole bunch of tiny little kind of dots, and that's exactly what I'm seeing under this. There's kind of a bunch of like amorphous blobs. They kind of look like a blob of jelly almost. So once you get one centered in your microscope, try to move your slide around to get one of those dots right centered in the microscope, and then you're going to turn it to the next power. Okay, so we're going to turn it to the next power, just like that, and we're going to look through. And there you go, you can see your own cells. So what they're going to look like is they're going to look like these kind of amorphous blobs, and they're going to have a darker dot in the center of them. So this is quite different from plant cells, right? They were these really kind of rectangular blocks that had these really dark edges around them. Those dark edges are because of the cell wall. When you look at a plant, it's kind of this amorphous blob because we don't have cell walls, right? When we cut ourselves, does it crunch like a celery? No, it doesn't, right? Because we don't have a cell wall. Cell walls are what makes plants rigid. It's what makes trees so, dense, and it's what makes celery crunch and break. We don't have that. So we don't have structure to our cells like plants do. So when you look at cells from an animal under a microscope, it's just going to kind of look like a blob. That blob is the cell membrane, and that's what contains all the bits of a cell. That dark spot in it is actually the nucleus, and the nucleus is what contains all of our genetic material. And our genetic material is called DNA, or deoxyribonucleic acid, which is quite a mouthful, but it's what codes for everything that we are. It's what codes for how we look, what color our hair is, what color our eyes are, how tall we are. Everything about ourselves, even the kinds of diseases that we might be susceptible to, genetics and DNA is what controls that, basically, to an extent. So this is where we're going with this. That's what holds our DNA, and the cells in our body are basically like holding us together, right? Obviously, we're all made of cells. So every single part of our body is made of cells, and there's different cells, that make up our liver. There's different cells that make up our brain and our bones. So have a look at that. Make your slides. See if it works, and if you have any issues or anything, just put them in the comments below. I'd love to hear your feedback, and I'll see you guys next week when we talk about DNA a little bit more.